Okay, so our next topic for the next couple of classes is to talk about how cells communicate with each other, which is a humongously important idea because cells are pretty much constantly talking to each other, as we'll see. And especially in certain organ systems, this is incredibly important. Wonderful example that leaps to mind is immunology. So in your immune system, all of your various types of immune cells have to talk to each other pretty much constantly in order to provoke an appropriate immune response against some kind of infection is a good example among many. Okay, so we're going to look at some ways in which cells can talk to each other. So there are sort of two broad ideas in that in some cases the sort of conversation that's happening is, to use sort of the older terms, long distance, where the signaling molecule, the one who's trying to talk, trying to say something, secretes usually what's referred to as a hormone that will circulate through the circulatory system and talk to another cell far away from that first cell. So this is, I don't know, in the brain, and the molecule travels through the circulatory system and has an effect, I don't know, in the feet. I don't know, just some silly example there. Okay, so long distance is usually referred to, the signaling molecules are typically referred to as hormones. But then there is also several types of more local phone calls, if you will. And these signaling molecules are usually not referred to as hormones. That term is usually used only for molecules that circulate through the circulatory system. But these local mediators also need to traverse a much smaller distance between the signaling cell and the target cell. So we'll look at those. So your book sort of defines 3 or 4 types or modes of cell communication. So it's worth kind of going through these just for the vocabulary. So the first type that we were calling here the hormonal type is usually referred to as endocrine signaling. So this is your typical, again, hormone, where the signaling cell, the one that's making the hormone, and the cell that it's talking to are far apart. Okay, so that the signaling molecule, the hormone, has to travel through the bloodstream to get from its source to its target. So that's endocrine signaling. The other types are more sort of localized, and so these are more like the second type here. And these sort of local phone calls are called paracrine signaling, where, again, the signaling molecule, or the signaling cell, and the target cell are not that far away. Okay, so we're not talking a hormone. The signaling molecule does not have to travel through the bloodstream. It just needs to diffuse over to reach the, relatively speaking, nearby cell. So that's called paracrine signaling. And your book takes neuronal signaling, puts it in a separate category. I don't know really why. Neuronal signaling, for me, is an example of paracrine signaling. I don't think it really merits a separate category, personally. So these two, for me, kind of go together. So neuronal is just an example of paracrine. And we will look at this particular example because it's a very good example of paracrine signaling. So we're going to count that one, these two, as sort of the same thing. So the third one for us is going to be contact-dependent signaling. In the first two, the signaling molecule is secreted, and actually in the fourth one that we'll talk about, it will be secreted as well. In contact-dependent signaling, as you can kind of get the feel here, the signaling molecule, whatever it is, usually in this case a protein, is embedded in the membrane of the signaling cell, as is the receptor in the target cell. So here there's nothing being secreted between the two cells. The two cells actually have to make physical contact in order for that particular signal to be achieved. There are a number of these examples like this in the immune system. So if any of you are taking immunology with me in the spring, we'll see examples of that kind of signaling as well. So, for example, between let's say the T helper cell and the T cytotoxic cell is a good example of contact dependence. So there are a handful of these where these two cells have to make physical contact with each other because that signaling molecule is not secreted. So that's the third type. Okay, so that we're going to extend our kind of analogy. So endocrine is sort of long distance phone calls, paracrine is local phone calls, contact dependent. Not a perfect analogy, but is sort of maybe face-to-face talking, if you want to extend the analogy. And then the fourth type, which I don't have a picture of here, is called autocrine signaling. And again, to extend the metaphor, this is sort of talking to yourself, where the signaling cell and the target cell are the same cell. So in this situation, this one cell secretes a signaling molecule for which it also has the receptor. So that's autocrine signaling. The one cell is both the signaler and the target cell sort of simultaneously. And again, there are good examples of this within the immune system as well. So, like I said, if any of you are taking that course with me in the spring, we'll see lots of signaling types of examples in that course, because in order for the immune system to work effectively, those cells have to be in sort of constant contact with each other. Okay, so there are multiple modes of communication. Again, this is more vocabulary than anything else. The four types kind of make sense? Okay. So we're going to spend most of our time in this course looking at examples of endocrine and paracrine signaling, but the other two are important in certain circumstances as well. This slide and the table sort of summarizes the types of molecules that we find acting as these signaling molecules. So the endocrine hormones, the ones that have to travel through the bloodstream, are basically of two types. They're either amino acids, derivatives, or small proteins, or they're steroids. They're steroid hormones. If you remember all the way back to probably either late August or early September when we were doing lipids, we looked at cholesterol derivatives that acted as steroid hormones, the sex hormones, corticosteroids, things like that. If you have no recollection of what I'm talking about, you might want to go back to that PowerPoint and look those up. We'll do a brief example of a steroid situation a little bit later in the presentation. So basically, either individual amino acids or small peptides, or cholesterol-derived steroid hormones. And then the same basic idea for paracrine signaling molecules as well. They're either amino acid derivatives or small lipids as well. So those are your two big categories, are sort of the protein side, protein types of hormones or signaling molecules, and the lipid-based signaling molecules. In sort of the broad theme, they both are going to do the same thing, but we'll see for those signals that are lipid-based, their receptors, as we'll see a little bit later, are usually not stuck in the membrane. They're usually floating around in the cytoplasm, and we'll see why that's so when we get to that part. Okay, this slide is showing another important sort of overall theme to cell signaling, which is, and using acetylcholine as our example, is that there's not actually like a zillion different signaling molecules out there. There's actually relatively a small number of different signaling molecules. Acetylcholine is one example. So it's a small amino acid derivative here. But it can have different effects in different cells, and that's what they're trying to show us in this top part of the figure. So this same signaling molecule, depending on what the target cell is, gets you different responses. So if you expose heart muscle cells to acetylcholine, they decrease their rate and force of contraction. They sort of relax. However, if you expose acetylcholine to skeletal muscle cells, they contract. So you get actually very different responses from two different kinds of muscle cells, even though it's the same signaling molecule. And then if you expose acetylcholine to, say, salivary gland cells and other types of secretory cells, they tend to cause secretion, which we just looked at before of break. So this idea of using the same small number of signal molecules, but eliciting different responses in different cell types is an important idea. So rather than have an absolutely completely different signal for every different possible outcome, it works really with a small handful of these signal molecules. And what's going to make the response different in each cell is the later part of the process. That's usually called the signal transduction cascade. So in general, we have the signaling molecule binding to some molecule. That is usually in the membrane, although when we get to the steroids, we'll see that that's not always true. And then this receptor is going to be connected to some signal transduction cascade that is going to then elicit some response. Relaxation, secretion, contraction, whatever. So the difference in each of these cells is not the signal. It's all acetylcholine. It could be part of the receptor, okay, that's making the difference, because presumably in each cell type, the acetylcholine receptor is going to be linked to a different signal transduction cascade, a series of steps that we'll see that gets you to the overall outcome. So this is the part that we're going to sort of focus on with a small number of examples, because you can actually take an entire course on this topic. There's an entire course called Signal Transduction on the books. It's a 400-level course. We don't teach it here. But if you go up to UPark, you can take an entire course in this topic. So it's just one signal transduction cascade after another. So we'll look at a small handful just to give you an idea of how this sort of works. So this general idea is sometimes referred to as pleiotropy, where you can get multiple effects. That's what pleo means. Pleo also means multi, so you can get many different effects from the same signal molecule. But it's usually in different cell types, as we're seeing here with the acetylcholine. And there are many different types of signals out there. So cells receive these signals sort of all the time. And this figure from your book is trying to demonstrate that. So cells, and we don't often think about this, but cells actually have to constantly be told to live. Those are called survival signals. And if those are shut off, that cell dies. Okay, so if it isn't constantly sort of tapped on the shoulder, stay alive, stay alive, stay alive, stay alive, stay alive, if that stops, the cell dies. Not immediately, but eventually, through a process called apoptosis, which unfortunately we don't have enough time left in the semester to look at, but if you take other courses like immunology, or if you take my cancer course, we talk about apoptosis there, so you can fill that in. But without that constant little tap on the shoulder, cells go through this process called apoptosis, which is essentially a suicide scheme. They kill themselves. So these survival signals are pretty important, right? Because you don't want your liver to kill itself anytime soon, or any other part of your body. So that's an interesting aspect of this, that they're constantly being told to survive. Many of them are also being relatively constantly told to stay differentiated, meaning stay a liver cell, stay a liver cell, more on the side, stay a liver cell, stay a liver cell, stay a liver cell, rather than become something else. Okay, so those are differentiation signals. And those, of course, are also important during embryonic development, right? Because we all start as a single fertilized egg, but then we eventually develop lung cells and liver cells and skin cells, etc. Those are all different differentiation signals. Again, I'm using all this immunology because I'm preparing to teach that in the spring, but that's an important part of the immune system as well. There are a couple dozen different kinds of immune cells out there, each of which differentiate from what are called hematopoietic stem cells in your bone marrow. And so they are getting slightly different, or each type is getting a slightly different set of signals, so that some differentiate into B cells, and some differentiate into mast cells, and others differentiate into T cells. And they're all getting slightly different little nudges to differentiate in a specific way. And then what we'll be looking at late this week and early next week are cell division signals. Okay, so occasionally, well, for some a lot, but others occasionally will get the signal to divide, right, through mitosis, which we'll start looking at on Friday. Some get that signal kind of all the time. Your stomach cells get that signal twice a day. Your liver cells only get that signal about once a year. Okay, so cells under normal circumstances only divide when they're told to. That is to distinguish them quite a bit from cancer, which doesn't need that signal anymore. Cancer cells divide out of control. They divide on their own without these external signals. So that's a big problem. So normally, cells only divide when they're told to. Cancer, the pathologic state, gets around that in a variety of ways. So there are lots of different signals out there. These are just a handful of sort of the common ones. We'll be looking at some more specific examples of sort of more mechanistic kinds of signals to do a particular task. So this is in addition to these kinds of things to secrete something, to contract, what have you. And so cells are getting lots of different signals all the time. Is sort of the theme here. And this is what I was kind of drawing over here. This is sort of the general scheme for how this works. We have some kind of signal molecule. We have some kind of receptor that hears that signal and then transduces that through a series of usually proteins, some of which will be enzymes, that create this signal transduction cascade that will lead in different cells to different results. So we'll look at one where the metabolism will be altered. We're going to look at the fight or flight response, which is basically altered metabolism type of thing. In lots of situations, these cascades lead to some kind of change in gene expression. So some genes are turned on, some genes are turned off, and that might mean the cell divides, for example. In some cases, it's going to tell cells to start moving, through rearrangements of the cytoskeleton that we've already looked at. So if you start changing around the actin filaments, for example, you can get a cell that's just sitting there start moving. Okay, so that could be another outcome as well. So depending on the specific signal, the specific receptor, the specific signal transduction cascade, you can get different results or different behavior changes in different cells. So like I said, we're going to look at several specific examples based on different types of receptors, and we'll look at several different types of behavior changes in the cell. So again, just sort of the same idea from a different book. The signal molecule is sometimes referred to as a ligand, sometimes as a primary messenger to distinguish it from some things that can happen inside the cell. But we're going to focus on this kind of example for the most part, where you have a receptor molecule in the cell membrane. You have an extracellular signal, binds to the receptor, clicks on in signal transduction pathways, some of which might include what are called second messengers. We'll look at all that when we get to specific examples that will create some kind of cellular response, perhaps some kind of change in gene expression. It will depend on the specific example. But that's sort of the general scheme for what we're going to kind of look at. Motion.
cell-to-cell signaling. Is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
So that's one type, right, where we change gene expression, we turn a specific gene on. Other examples would not include the transcription factor. It would just all, say, be in the cytoplasm and something else would change. But just to give you a general sense of how this is going to work. And we can sort of organize the topic by differences in what the receptor protein looks like. So again, we're going to be focused on this in this early part on receptors that are obviously proteins embedded in the membrane. So we've already studied how that happens, right? Stop transfer sequences, inserted that protein in the membrane of the ER, it traveled through the Golgi, eventually gets put into the membranes. We've studied all that already. Okay, so we know how they get in there. So different people, as they've been studying types of receptors, have sort of come up with this sort of three-category system. We're not going to look at them in exactly this order, but that's okay. We are going to start out with what are called ion channel-linked receptors. So these are receptors that are not only receptors for a signal, but also are ion channels. So typically, this is showing sort of how it typically works. So the signal molecule binds, and that typically opens up the channel, and that's going to let in a specific ion, a sodium ion, a calcium ion, whatever. And that change, or that entry of the specific ion, will have some specific effect on that target cell. Okay, so we're going to look at nerve cell propagation as an example of an ion channel-linked receptor. We're going to do these last. So the second type we're going to look at very briefly are enzyme-linked receptors. So these are receptors that are also enzymes. Okay, so usually their cytoplasmic side are typically have some kind of enzymatic activity or catalytic activity, often a kinase kind of activity, a phosphorylation activity, although there are others. So we'll look briefly at that. And then the third type that we will spend a good amount of time on are called G protein linked receptors, because these receptors, the receptors here in green, when that gets triggered by a signal molecule, it activates a type of protein called a G protein. And it's called a G protein because it interacts with the nucleotide GTP, so they're called G proteins for that reason. And as we'll see, an activated G protein can then turn on some other protein like an enzyme or something like that. So we'll look at a couple of examples of those as well. Okay, so we'll look at a couple specific examples for each of these types, but this is a nice way to sort of organize the topic is by sort of type of receptor. Okay, so we're going to start off with the ion channel linked receptors, and we're going to use nerve cells as a really, really good example of this type of system. So, if you haven't had anatomy or physiology in a while, just to remind you or teach you quickly about a nerve cell, you have sort of the cell body here where the nucleus are and little extensions called dendrites. And then, I think we've already looked at this already, the long section of the nerve cell that in some cases can be up to potentially a meter long is called the axon. Okay, what cytoskeletal element helps keep this axon like that? Not actin. All right, one down, two to go. Microtubules. All right. So this axon is maintained through long microtubules. And then you have at the far end of the cell something that's usually called the terminal knob, or here they're calling it the terminal bulb, sort of the same idea. And typically, this is where we're going to focus, here's where the signal molecule is going to come out. The next cell would be right next door, and we're going to see how that signal molecule will trigger an ion channel link receptor in the next cell. So this is an example of paracrine signaling if we want to link it back to those types as well. So what we're going to see is that the initial nerve signal is going to come down, reach the terminal knob, cause the secretion of the signal molecule into the little space between this nerve and the next nerve. That's called the synaptic cleft. Okay, so you've probably heard of neurotransmitters. Okay, so those are the signal molecules, and they're going to bind to, on the second membrane, ion channel linked receptors. So that's how we're going to look at this here. Okay, so this is actually a nice micrograph here, so you can see that the terminal knob is just filled with vesicles that have all of these little neurotransmitters contained in them. So these are just a couple of examples of various kinds of neurotransmitters. So acetylcholine that we looked at earlier is one of them, but dopamine you might be familiar with, serotonin, norepinephrine, so there are a number of different neurotransmitters out there. But they all work in a relatively similar overall way, so we're just going to look at neurotransmitters sort of in general, okay, but in case you want some specific examples. Okay, so here's the basic idea. Okay, so here's our first nerve, here's our second one, here's the synaptic cleft in between, and these purple things here are the ion channel linked receptor. So what basically happens, again, if you studied anatomy and physiology already, you already know this, but a nerve signal, the electrical part of this, is sort of a flood of sodium ions that rush down the length of this first nerve cell called the action potential. And so this first nerve cell gets activated, sodium rushes in, that stream or that flood of sodium rushes down the length of this cell and reaches the terminal knob. Now if you want to get a little bit more into detail, what that triggers, so that's action potential, is actually going to trigger the calcium channel to open, and in floods calcium, which will then cause these vesicles that have the neurotransmitters in them to secrete those neurotransmitters. Okay, so what kind of secretion would we call this? Two types of secretion that we looked at. Well, all secretion is exocytosis, but is this constitutive or is this regulated? Looks like you guys ate too much turkey and didn't do enough cell biology. Right, this is a regulated system. This only gets secreted. This only happens when you get that sodium signal. So it's a regulated type of situation. So we're making all kinds of connections. That's the wonderfulness of the end of the semester. Okay. So the sodium ions rush in or rush down the cell. They open up the calcium channel. Calcium comes in, causes the regulated secretion of those neurotransmitters, which will then flood into the synaptic cleft. There is a binding site on these ion channel linked receptors on the next cell. I'll show you a better picture of them in a second. But basically, when the neurotransmitters bind, they're going to open up those ion channel linked receptors on the next cell and start a new action potential. So these ion channels are specific for sodium. So sodium is going to rush in, that's the action potential. That triggers the next cell. So we basically change the signal from an electrical signal to a chemical signal back into an electrical signal in the next cell. So that's how the signal sort of jumps from one nerve cell to the next in the relay. And is across a chemical synapse here, as we would call it, through these neurotransmitters. This is probably more detail than we probably need, but if we want to get those cytoskeletal elements back in the mix here, of course, all of those little vesicles with the neurotransmitters in them are being walked along those microtubules. Remember we talked about the sort of microtubule highway before the break as well. So technically, they're being walked along. That's going to be triggered again by that calcium that's coming in. They're going to walk along, fuse with the membrane, and release their neurotransmitters, like acetylcholine. So we'll use that as the example here. Again, here's the ion channel link receptor on the second nerve cell. When acetylcholine binds, it opens it up and allows sodium to rush in, again, starting a new action potential. So it's kind of a nice, clever system to get two cells, or get a signal from one cell to the next that aren't actually touching. Right? So there is that physical gap, that synaptic cleft. So we have to get across that somehow, and we get across it through these neurotransmitters. So again, we change it from electrical to chemical, back into electrical signal in the next nerve cell. So I like this example a lot. It's a kind of a clever way of traversing that space between the two adjacent cells.
Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse? There is a narrow gap, the synaptic cleft, between the synaptic knob of a sending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. Having done its job, the neurotransmitter is broken down by an enzyme, and the ion channels close.
Okay, so a couple of important ideas from here. Again, we're looking at the specific situation where the second cell is being activated. Okay? But other neurotransmitters would turn the signal off in the next cell, so there are variations on this. We're just looking at this specific example. And then the other part of this that they mentioned right at the end is the off signal. Whenever we study signal transduction and cell communication, we're usually very, very interested in the on signal. How do we turn on this, the action potential in the next cell? How do we get the cell to start moving or whatever? But the off signal is also very important, because if this signal never turned off, you're still going to be feeling that stimulation from 3 years ago, right? That's not a good idea. So we need to be able to turn this off as well. Typically in synaptic clefts, there are enzymes that break down the neurotransmitters, and that's the off switch, right? So if we destroy those neurotransmitters that are floating out here, then they can't bind to the receptor anymore and keep the channels open. So there's always an off switch to any kind of cell communication event. We just don't often stress them quite enough, probably, when we're studying this. But always keep that in mind. There's always an off switch to these kinds of situations. Okay, so this is sort of the classic example of an ion channel linked receptor is in the nervous system. We will actually come across one other a little bit later, probably not till Wednesday. but we will see one other example of this as well, but this is, like I said, is sort of a classic example. So this is also an example of a paracrine kind of situation, right? Because the two cells, the signaling cell and the target cell, are really close together, okay, but not completely touching, so this would be paracrine type of signaling as well. This example makes sense, and ion channel link receptors, this type of receptor makes sense. Okay. Okay, the second type of channel, or I shouldn't say that, the second type of receptor is called either an enzyme-linked receptor, or sometimes they're just referred to as catalytic receptors, whichever you prefer. The overall idea here is that the receptor now is no longer an ion channel. It has some sort of enzymatic activity. And very, very often, not 100% of the time, but very, very often, that enzyme activity is a kinase activity. And I'm not sure how much we've talked about them in here, but kinases are incredibly important to this story. Kinases add phosphate groups to things. Typically to proteins, so we'll put to target proteins because that's usually what they do. And when you add phosphates to specific proteins, you often can turn them on. It's often the on switch. Although sometimes the off, but usually more often it's the on switch. And we're going to see this as a major, major theme in cell communication and these signal transduction pathways. A series of proteins gets phosphorylated in a specific order, and that turns them on, and they do something else, and we get this long line of things to do. So in the example of these catalytic receptors, typically it is the cytoplasmic portion of the protein, so you have an extracellular portion, a transmembrane portion, and a cytoplasmic section. It's very, very often the cytoplasmic side that has this kinase activity, this enzymatic activity. And as we can see here, the first and the third panels are very, very good examples of catalytic receptors that are part of what's called the sort of growth factor family of proteins. So this EGF stands for epidermal growth factor. PDGF stands for platelet-derived growth factor. There are a zillion types of growth factor kinds of receptors, and they are all of this catalytic type. So you see here this kinase domain. Specifically, and if we want to get a little bit of detail here, there are sort of two different kinds of kinase activities. One is called, as you see there, the tyrosine kinase type, and the other type are the threonine serine kinase type. And it just tells you what amino acid picks up the phosphate group. And so the tyrosine kinases obviously phosphorylate tyrosines, and the threonine serine type phosphorylate threonines and serines. If you remember all the way back to amino acids, all three of these types of amino acids have a hydroxyl group on their R group, and that's where the phosphate gets attached, is to that hydroxyl. So again, we can sort of keep dividing and subdividing these kinds of things. So there's a tyrosine kinase family, or sort of, I guess, subfamily, and a threonine serine subfamily. We don't have to worry about that too much, but I just wanted to point that out since they had put it there. And then the one in the middle is also a good example. Your insulin receptor, okay, that gets triggered by eating something with a lot of glucose, will then trigger the release of insulin from your pancreatic beta cells, which in turn, that insulin will bind to this receptor, typically in liver cells and muscle cells, which will then tell the cells, hey, we need to put some glucose transporters into our membrane so we can suck up all this glucose that you just ate from a candy bar or a piece of pumpkin pie after Thanksgiving dinner or whatever. That is also mediated through the insulin receptor, which is also a catalytic type of receptor. It's a tyrosine kinase receptor as well. So there are a large number of these types of receptors, these catalytic receptors around. So this gives you a little bit more detail. What typically happens here is that two of the receptors have to get together, so they dimerize. And when the two get together, that is what usually triggers the enzyme activity. And in this example, what happens is that the one half phosphorylates the other half. So all of these tyrosines pick up phosphate groups. And this is now the active version of the receptor, and that will trigger the rest of the signal transduction cascade, whatever it might be. But that's how it gets activated. And we're not going to look at a specific example for a catalytic receptor. We're just going to kind of look at it more in general. But what tends to happen after this, so after the receptor is activated by this phosphorylation, those phosphorylated tyrosines sort of act as little hooks for other proteins that are usually called, well, they're not calling it here, they're just calling them intracellular signaling proteins. These are sometimes referred to as relay proteins. Start to bind to specific tyrosines that get phosphorylated. So you can see that this light blue one hooks up to the top phosphorylated tyrosine. The tan one does the middle one. The dark blue does the bottom one. So there are specific places where these proteins bind, and then other proteins will start to bind to these relay proteins, and that starts this long process. So you don't need to memorize this one, but here's one example that if you take my cancer class, we study in some detail. So this is a pathway that will eventually lead to cell division. Okay, so here's the growth factor, because that's really what growth factors mean, right? When you throw a growth factor at a cell, the cell will grow, but then it divides. So it's a division type of signal. So it binds to the receptor, it dimerizes, that will activate the receptor, and then in turn, that activated receptor will activate this protein here called RAS, which is an extraordinarily important protein in terms of cell division. And if you study cancer to any extent, you find that this particular protein is in virtually every cancer known to humanity is mutated. Okay? And it's mutated in such a way that it's always on. So think about that. If this pathway, by triggering RAS and the rest of it, that we'll look at in a second, eventually gets you to cell division, if you're a cancer cell, and you can click that and lock it in the on position, what does that mean for the cell? What is the cell now always going to be able to do? Divide, which is the basic definition of cancer. And so that's another important reason why we studied some of these pathways, is because some of them can have pieces of them changed, mutated, clicked permanently in the on position. That allows those cells to divide all the time without being told to. Okay, so if we click this one on, we don't need that signal anymore, do we? Because this part downstream is always on. So that's how cancer cells get around the need to be told to divide. If you click that on, and really any of these others as well, but RAS is a very, very common one. This is always on, because that cell can always divide without the need for that growth factor anymore. So that's, again, a hallmark of cancer. But this is also showing us how these cascades sort of build. So we have the growth factor binding to the receptor is activated, it activates RAS, which in turn hooks up with this other protein called RAF, which then activates MEK, which then activates ERK, and a few more steps after that to eventually tell the cell, hey, divide please, and go through mitosis.
I have a side question. You said in cancer, RAS causes the cancer cells to divide more often?
If it's mutated, yes.
So would this chemotherapy or radiation try to turn that off?
No, it kills the cell that's dividing too rapidly.
Okay.
And that is why typically those things have pretty nasty side effects, because they can't just target the tumor, because other cells in your body are also rapidly dividing, like your stomach cells. They divide twice a day. Your hair follicles, Okay, so here's our side effects. So those same things, the radiation, the chemo, also, while killing the cancer cells, hopefully, are also killing off your stomach cells to get nauseous. They start killing your hair follicles, so your hair falls out. They start killing off your cells in your bone marrow that make your red blood cells, so you become anemic. So we can track all of those side effects to exactly how those things work, because they're not specific. Well, they're specific only for actively dividing cells. So any actively dividing cells get hit by those things. So that's where we're trying to go with cancer treatment, is to not do this, non-specific thing. We're trying to focus on the tumor to, greater or lesser success. But yeah, that's a good question. Yeah, so it's not looking at that specifically in that sense. Some of the newer technologies, some of the newer drugs are trying to do that, so that, yeah, they are targeting mutant RAS, which should only be in those cancer cells, and they should leave your stomach cells and your hair cells and those things alone, ideally. So that's what people are trying to do. So, hopefully a little bit of incentive to study this, because this really plays a huge, huge role in cancer biology. So here's a little bit more on the RAS pathway. Again, you don't need to memorize this particular one, although I do want you to know the nerve cell one in detail. These you don't need to know in detail. Okay, but again, it's the same idea, and again, we're just building a large pathway. So the signal molecule binds to the receptor. The receptor, in this case, activates RAS, which in this example then activates MAPK, which turns on specific genes through AP1, while at the same time it's activating this other pathway called GRIB2 and SOS, and activates this other protein, PLC gamma, which breaks down that particular lipid in the membrane, which opens up calcium channels. So one signal can have a very, very large effect on the cell. But again, these enzymatic examples you don't need to worry about in any detail. Okay, this is probably a good place to stop. Okay, so we'll pick up with G protein-linked receptors, our third type, on Wednesday. We'll look at steroid hormone receptors, and that should finish up this topic. And then we'll get into mitosis and meiosis to finish up the course. Okay. Please take a look at this stuff.